Imagine a world where curing a genetic disease is as routine as replacing a broken part in a machine. A single injection, guided by artificial intelligence, could seek out the flawed DNA, repair it with near-perfect precision, and leave behind no trace of error. This isn't science fiction anymore, it's the unfolding reality of CRISPR 3.0, which marks a new era of gene editing, supercharged by AI. And the implications stretch far beyond medicine. They reach into agriculture, synthetic biology, climate resilience, and even the ethical foundations of what it means to be human. But here's the twist. The technology isn't just evolving incrementally. With AI in the driver's seat, the rate of progress is accelerating exponentially, much like how GPS turned ordinary maps into a real-time global navigation system. CRISPR on its own was a revolution. CRISPR guided by AI could be the moment biology becomes programmable at scale. Subscribe, like, and turn on notifications if you want to stay ahead of the breakthroughs shaping our future, because this story could redefine life itself. To understand why this matters, let's step back. Traditional CRISPR, discovered in 2012, gave scientists molecular scissors that could cut DNA at specific spots. It was like having a word processor for biology, but with one huge catch: accuracy. The first generation of CRISPR had an error rate of up to 10%. That doesn't sound like much, until you realize even a single off-target cut could cause unintended mutations, turning potential cures into new risks. CRISPR 2.0 brought refinements like base editing and prime editing, which worked like swapping out individual letters instead of tearing out whole words. Accuracy improved significantly, with some labs reporting error rates dropping below 1%. But now, with CRISPR 3.0, AI is being woven into the process, and the difference is dramatic. According to recent studies, machine learning models can predict off-target effects with 90–95% accuracy and optimize guide RNAs that increase precision by up to 50%. In practical terms, what once took months of trial and error in the lab can now be compressed into days or even hours of AI-driven computation. Imagine the same leap in speed and precision as moving from dial-up internet to 5G. That's the magnitude of the shift CRISPR is experiencing right now. Here's how it works. AI systems, trained on vast datasets of gene editing experiments, learn the hidden rules of DNA folding, protein interactions, and repair pathways, patterns invisible to human intuition. When scientists feed in a genetic target, the AI doesn't just spit out a single guide sequence. It runs millions of simulations in silico, ranking the safest and most effective candidates. It's the equivalent of an AI chess engine that can see 20 moves ahead. Only instead of checkmates, the goal is flawless DNA repair. To make this more relatable, picture trying to repair a damaged sentence in a massive library of books. A human might search for hours, flipping pages to find the error. An AI, however, reads the entire library in seconds, not only spotting the flaw, but also predicting the best way to correct it without disturbing the surrounding text. That's the leap in intelligence CRISPR 3.0 brings to biology. We're not yet at 100% perfection. Even with AI, error rates still exist, and the technology is primarily confined to lab and preclinical trials. But the progress curve suggests that by early in the 2030s, with 80–90% confidence, AI-guided CRISPR could reach clinical-grade reliability suitable for routine therapies. So what does that mean in human terms? It means that diseases caused by single-gene mutations like sickle cell anemia, cystic fibrosis, or Huntington's disease could move from manageable but incurable to fixable at the root. In fact, in December 2023, the FDA approved the first CRISPR-based therapy for sickle cell disease. Now, with AI enhancing precision, the cost and scalability of such treatments could drop by an order of magnitude. If the first therapy costs $2 million per patient, projections suggest AI-guided CRISPR could push that below $200,000 by 2030, and possibly under $50,000 by the mid-2030s. That's the difference between a moonshot cure for a few and an accessible treatment for millions. Think about this. What vaccines did for infectious disease in the 20th century, AI-guided gene editing could do for inherited disease in the 21st. Now imagine a global healthcare landscape reshaped by such accessibility. A child in rural India with a life-threatening genetic condition could receive the same cutting-edge treatment as someone in a state-of-the-art hospital in New York. This democratization of healthcare could shrink global health disparities, unlocking human potential in regions historically constrained by preventable disease. But medicine is only one arena. Agriculture stands to be transformed as well. AI-enhanced CRISPR is already being used to design crops that are more resilient to drought, resistant to pests, and even fortified with extra nutrients. Instead of the blunt force methods of GMOs from the 1990s, which often involved inserting foreign genes, CRISPR 3.0 allows subtle, targeted tweaks. For example, scientists are editing the genomes of rice plants to increase yields by 15–20% while reducing water usage by up to 30%. That's not just a scientific victory, it's a lifeline for a planet facing climate stress and food insecurity. And then there's synthetic biology, the field where life is treated like programmable hardware. Picture designing microorganisms that can eat plastic waste, generate clean biofuels, or produce rare medicines on demand. With AI predicting and optimizing edits, this could shift from proof-of-concept experiments to industrial-scale reality within a decade. By 2035, with about 70% confidence, experts anticipate AI-optimized microbes could supply up to 10% of global fuel needs, cutting billions of tons of CO2 emissions annually. Think about the possibility of future cities powered not by fossil fuels, but by organisms designed to convert waste into energy on demand. In that scenario, garbage trucks wouldn't just haul waste, they'd be feeding bioreactors that power entire neighborhoods. Of course, with power comes risk. Ethical debates are heating up. The question is no longer can we edit life, but should we, and under what rules? The specter of designer babies looms large. AI makes it technically easier to select or enhance traits in embryos, like intelligence, height, or resistance to disease. But should society draw a line between curing illness and enhancing beyond the human norm? And who gets to decide? While embryo editing grabs headlines, in reality, regulations are extremely tight. Most countries ban germline editing that would pass changes to future generations. Current applications are almost entirely therapeutic, not enhancement. But as AI makes the process safer and cheaper, the social pressure to push those boundaries will only grow. Imagine parents browsing a catalog, not for shoes or furniture, but for potential genetic profiles of their children. That's the kind of slippery slope ethicists are warning about, and why the debate must keep pace with the technology. Interestingly, AI may also help address the ethics. By modeling long-term genetic outcomes, AI could highlight risks humans might overlook, such as subtle trade-offs between enhanced traits and hidden vulnerabilities. In this sense, AI is not just a scalpel but a safety net. Let's ground this further with a timeline. By 2027–2028, with 75% confidence, we'll see AI-enhanced CRISPR therapies expanding beyond rare diseases into more common conditions like heart disease or diabetes, where genetics play a contributing role. By 2030–2032, with 85% confidence, agricultural applications could be widespread, with climate-resilient crops becoming the norm in regions hardest hit by drought. And by the mid-2030s, the fusion of AI and CRISPR and synthetic biology could create new industries we haven't yet imagined. Industries as transformative as the semiconductor or internet revolutions of the past century. And yet, we must remember the complexity of biology. Delivering CRISPR tools into the right cells of the human body remains a technical hurdle. Viral vectors, nanoparticles, and other carriers are still being refined. Off-target effects, while greatly reduced, aren't eliminated. And the complexity of traits influenced by dozens or hundreds of genes, like intelligence or athletic performance, means we're far from editing them reliably. The reality is that CRISPR 3.0 is a powerful scalpel, but it's not a magic wand. Think of it like autopilot on a plane. AI can make the journey smoother and safer, but you still need skilled pilots and regulations to guide the flight. The societal impact could be staggering. Imagine a future where no child is born with preventable genetic disease, where crops are designed for each climate zone, reducing hunger worldwide, where synthetic organisms clean up oil spills or generate carbon-neutral fuels. At the same time, imagine the debates over fairness, access, and human identity. Will these breakthroughs deepen inequalities if only the wealthy can afford them? Or will they become the great equalizer, offering health and resilience across the globe? If you want to be part of these conversations at the cutting edge of science, subscribe, like, and turn on notifications. As the science advances, the role of storytelling and public dialogue becomes crucial. Technologies don't exist in a vacuum. They live in the social contracts we create around them. Just as nuclear power was shaped by treaties and regulations, CRISPR 3.0 will demand frameworks that balance innovation with caution, hope with humility. Consider how similar paradigm-shifting technologies were handled historically. When electricity spread across the globe, it wasn't just about wires and bulbs, it redefined human activity, industry, and culture. The same holds true here. CRISPR 3.0 isn't merely a laboratory breakthrough. It's a force that could reshape human civilization itself. What's striking is how quickly the science fiction future is becoming science fact. 10 years ago, gene editing was an experimental curiosity. Today, it's curing real patients. 10 years from now, with AI accelerating discovery, we could be living in a world where biology is as editable as software. The question is not whether that world is coming, but how prepared we'll be when it arrives. Remember how the internet went from obscure academic networks in the 1980s to shaping every aspect of life by the 2000s? That's the trajectory gene editing could follow in the next 2 decades, only faster. And so we arrive at the final question: where does this all lead? The horizon of CRISPR 3.0 stretches far beyond what we can see. Some scientists whisper about CRISPR 4.0, where gene editing doesn't just fix DNA, but dynamically rewrites it in real time, like a living operating system. Others talk about hybridizing AI with biology so deeply that we blur the line between natural evolution and guided design. These possibilities may seem distant, but so did editing the human genome just 15 years ago. Most of these visions remain speculative. We don't yet have the control, nor the ethical consensus, to take such leaps. But the direction of travel is clear. AI is no longer just helping us understand life, it's giving us the tools to rewrite it. So what do you think? Should AI-guided CRISPR be used only to cure disease, or is it inevitable we'll move into enhancement and design? Leave your thoughts in the comments. If you want to stay on the frontier of how technology is reshaping humanity, subscribe, like, and turn on notifications. This is Tech Mythos.